All right, thanks, Melanie. Thanks for the introduction. Thanks for inviting me. It's cool and perplexing to be giving a talk back at my former PhD department. And it's a real shame that I can't be there in person, because it would be nice to see a lot of you again and just kind of hang out and talk about science and other things as well. So I'll be talking today about something that I've become interested in since starting at the University of Toronto, which is the evolution of temperature-dependent sex determination and its ecological implications under climate change. Now, this is something that both Melanie and I jumped into together, and we started studying it a few years ago. And then Melanie's since, of course, moved on. Because when I used to think about sex determining mechanisms, I'd always default to what could be called, in my world at least, is boring old XY sex determination, where we have a sperm with a particular genetic complement that fertilizes an egg with a particular genetic complement. There's sex chromosomes involved. If the sperm's carrying the Y chromosome, then the embryo becomes male, and if the sperm's holding the X chromosome, then it becomes female. And this is a system of genetic sex determination that pervades among mammals. But it's not the only system of sex determination, genetic sex determination. There's also the ZW system of sex determination, which completely turns the XY system on its head. So here, under ZW, it's the female that is the heterogametic sex. So she is ZW, and the males are ZZ, the homogametic sex. And when you look at some of the other vertebrate clades, what you have is a diversity of sex-determining mechanisms among the different species in that clade. So, for example, among fishes, we have the XY system of genetic sex determination. We have the ZW system of genetic sex determination. And we also have environmental sex determination. So that's where, whether or not the species has sex chromosomes or not, sex is determined during embryonic development by an environmental factor. And so the most common form of environmental sex determination is temperature-dependent sex determination, which was only discovered in 1966 by Madeleine Charnier from the University of Dakar in Senegal. And so some fishes do have TSD, temperature-dependent sex determination. I believe they're Atlantic silversides and maybe some related species, but by and large, most species with TSD are reptiles. So here's a species level reptile phylogeny. In light green at the tips of the phylogeny are species with genetic sex determination. So you can see there's a lot of genetic sex determination in snakes and lizards. And in dark green are species with temperature-dependent sex determination. So all crocodiles that we know of, and most turtles, and also the tuatara, which is, of course, in its own order of reptiles. TSD is ancestral, so it evolved, at least 320 million years ago or so. So the theory is, at least, it's been lost in most squamates, which are snakes and lizards. In fact, no snakes have TSD. It's only lizards that do. And it's been retained, TSD has been retained in crocodiles and in turtles. And so most of us don't actually care about temperature-dependent sex determination. When we hear about temperature-dependent sex determination, we only hear about it in the context of, well, populations of turtles or crocodiles or whatever is going to go extinct because they're all going to become one sex under rapid climate change. Maybe that's true. Maybe, we don't know. But we also have to appreciate that one of the great mysteries of TSD is that it persists through deep time. So TSD evolved, maybe it's been around at least for about 300 million years. So we're looking over here on the left of my screen. And if you look at the climatic change that's happened during that time, it's been absolutely massive. So even if I'm wrong about when TSD evolved by like 150 million years, it doesn't matter. The argument is the same. There's been massive climatic change in the last few hundred million years. So how is TSD persisting through this? We don't actually know the answer to this. So this is one really cool reason to study TSD, both in terms of deep time and what's going to happen in the contemporary climate. And the other really cool thing about TSD is we don't even know what its adaptive significance is. We do not know why TSD evolved, or at least we can't agree. Different people have different ideas, but no one has this kind of silver bullet that explains the evolution and maintenance of TSD. So as a early career assistant professor who's interested in kind of ectothermic vertebrates and thermal ecology and global change, TSD is of course kind of it's inexorable that I'm going to be drawn to this topic. And so today, what I want to talk about is I'm going to give you a quick overview of how TSD works. Then I want to talk mainly, I think, about why I think TSD evolved in reptiles. And then I'm going to talk briefly about a 50-year study that I'm helping run up in Algonquin Park that kind of hints at some climate change resilience in a population of turtles up there, a species with TSD. All right, so how does TSD work? So as an embryo develops, it goes through a series of ontogenetic stages. Stage 1, let's say, to stage 20. We can score and make up these stages however we want, based on characters or whatever. And there's a particular series of these ontogenetic stages that occur about in the middle of development, stages 6 through 11, and according to Eintema, where sex is labile, where if you kind of crank up the temperature, you'll get a certain sex, or if you lower the temperature, they'll all become a different sex. So there's this thermosensitive period, and that's when the gonads are differentiating, and that's when the embryo is its sex is sensitive to temperature. So if you manipulate temperature after or before the thermosensitive period, it won't affect sex, only during the thermosensitive period. And so one way of thinking about how it works too is if we have a constant temperature experiment set up. So we have a series of, let's say, 5 or 6 or 7 incubators, and we have a different temperature in each incubator, and we put, say, 50 eggs in each one of these incubators and let the eggs incubate throughout development. And then at the end, we get the sex ratio of the hatchlings, which might be 100% male, 0% male, or 50% male, and so on and so forth. So the proportion of the clutch that is male is on the y-axis with 100% or 1 at the top and 0 at the bottom, representing female bias clutches. So what the temperature sex reaction norm might look like is something like this. So each one of these black dots would be data from one incubator, and then we've overlain a best fit reaction norm that describes the relationship between constant temperature incubation and the probability that, or the proportion that the clutch that is male. A couple key parameters that we've kind of made up, but that, you know, hypothetically, I suppose, exist, are the pivotal temperatures. So in this case, always the pivotal temperature is the temperature where you're expected to get a 1-to-1 sex ratio. So in this case, there's 2 pivotal temperatures because we have a Gaussian-shaped reaction norm. So we have a pivotal temperature at about 21 degrees Celsius here, roughly, and another pivotal temperature at about just under 27.5 degrees Celsius here. All right. So it's not necessarily the case that we're going to have a Gaussian-shaped relationship or reaction norm for sex and temperature. What I've just described in the previous slide was the FMF pattern, where we have females at cool temperature, females at warm temperatures, and males at intermediate temperatures. This is supposed to be the ancestral condition. And then we have derived conditions, or supposedly derived conditions, where we have an MF reaction norm, where males arise at cool temperatures and females arise at warm temperatures. And then, of course, we have the opposite of that, which is females arise at cool temperatures and males arise at warm temperatures. So it runs the complete gamut of possibilities. And you can see that maybe FMF is ancestral because it tends to be in ancient clades, and also because it subsumes the MF and the FM curves. But given all these different temperature by sex reaction norms, you can imagine why it might be difficult to find a unifying explanation for the adaptive significance of TSD. If we wanted to, we could overlay our pivotal temperatures on each one of these reaction norms, and you could see that they'd kind of just be in the middle of each one of these curves, with the FMF curve being the one that has 2 pivotal temperatures. All right, so that's a brief overview of how TSD works, but what is the adaptive significance of TSD in the wild? So we do have a general model of how, of why temperature-dependent sex determination might be adaptive, or how it might be adaptive rather. So TSD can be adaptive when incubation temperature influences fitness in a sex-specific manner. So in other words, if we have, lifetime reproductive success, so fitness on the y-axis, incubation temperature here on the x-axis, this particular pattern of reaction norms for fitness could favor the evolution of TSD, and that's because here we have males that are completely unaffected, their fitness is unaffected by incubation temperature, but for some reason, female fitness increases with incubation temperature. Maybe warmer females grow faster and they have more eggs or something like that, right? So under this pattern of, under these fitness functions, we'd expect this temperature by sex reaction norm to evolve, where you'd get males at cool temperatures and females would be produced at warm temperatures. Remember, it's percent male on the y-axis here. So high percent male at low temperatures, low percent male at warm temperatures. Alternatively, I mean, you could have something that's completely opposite, right? You could have kind of males fitness increasing weekly with temperature and female fitness decreasing radically with temperature. In that case, you would be this temperature sex reaction norm that might be favored, where you have females produced at cool temperature and males produced at warm temperature. And so this is kind of getting at what the problem is. We don't have a specific explanation within this Charnov-Bull framework that broadly explains why the fitness of males and females is differentially affected by incubation temperature. Sure, we can make up explanations like on a species-specific basis that fall into the Charnov-Bull framework, but really probably it would be much more convincing if we had kind of a broad hypothesis that applied to many, many species that explains why there's a temperature by sex interaction for temperature by sex interaction for fitness. And that's what I'm going to be looking for today. And so what I'd argue is that part of the confusion is that we're actually dealing with two different clades here with two different patterns of sex determination. So if you look at the phylogeny, it seems pretty obvious to me that what we have here is largely a loss of TSD over time in lizards and snakes. And the tuatara has happened to retain TSD. So the tuatara and the squamates, which are all these guys, are a sister group. Whereas the crocodilians and the turtles are more closely related to each other than they are to the squamates and the tuatara, and they've mostly retained TSD. So really this is a tale of two different clades. So what I'm going to do is I'm going to try and explain TSD or justify why we might only want to explain the adaptive significance of TSD for each one of these clades separately. So you can see that most turtles and chelonians, turtles and crocodilians rather, have retained TSD. They also happen to have really similar life histories. They tend to mature at a relatively late age and they live forever. Whereas squamates, at least, they mature relatively early and they don't live very long. So we have completely different life histories in squamates and in crocodilians. So not only that, not only do we have kind of independent evolution of TSD in these different groups over a long period of time, large differences in life history, we also have differences in the temperature sex reaction norm that are kind of broadly conserved within these different clades. So in turtles, we have FMF and MF, and in crocodiles, we have FMF. Turtles are the only group to have MF. Whereas in squamates and the tuatara, they have the FM system as well as that ancestral condition as well. So given the independent evolution of TSD, or like the long periods of independent evolution of TSD in these groups, the differences in life history and the differences in the temperature sex reaction norm, I suspect that we're probably safe in invoking an adaptive explanation that applies broadly to turtles and crocodilians, and not having to worry about having the same explanation apply to squamates, because in squamates, I'm fairly convinced something different is going on. And we can talk about that in the question and answer period if you want, what that explanation might be. So here, the adaptive explanation I'm going to give you broadly will invoke a temperature by sex interaction for fitness for the FMF and MF patterns of TSD. So for crocodilians and for turtles. And the explanation is rooted in classic principles of evolutionary ecology. So when we think about reproductive success in a breeding population of adults, so we're not talking about lifetime reproductive success anymore, we're talking about breeding success, reproductive success in a breeding population of adults. What we expect is that male fitness is going to be limited by the number of individuals, number of females that they can mate with. It'll be exponentially distributed with a modal value here of 0. So most males get to mate with nobody, and a few males get to mate with a whole bunch of different females. On the other hand, female fitness is limited by how many eggs they can produce by fecundity. It's not mated, or it's not limited by the number of mates they can have. So female fitness will be normally distributed with a modal value well above 0. And so you can easily imagine this a system in a mammalian system with harem guarding, like seals, which I hope this species is, because you guys are all marine biologists. In many mammals, you have males that guard large harems of females, and so it's easy to see how this male would exclude other males from mating and mate with a large number of females, and it's easy to see how we get the exponentially distributed reproductive success of males. But it's not just in mammals with harem guarding that we'd get that. So in the marine iguana, we see the same pattern. It's a territorial species without TSD, but we also see it in things like the rough-skinned newt. So this is a pervasive pattern that we see throughout the vertebrate kingdom, where we have a few males capturing the overwhelming majority of mating success. And it's not as if this is just, it's just a few lucky males that this is occurring to. Their phenotype is non-random with respect to mating success. It's high condition males that capture a disproportionate number of mates. And what I mean by condition is general health, vigor, how vigorous they are, fat reserves, how tough they are. It's a catch-all term to describe the quality of the male phenotype, or the quality of the phenotype in general. And we know that condition is important in terms of mating success and sexual selection. In fact, these secondary sexual characteristics that we're looking at, so the peacock's tail and the sage grouse and the colors on these lizards, are condition-dependent traits. When the males have extra energy, they channel them into these traits to advertise to the female saying, hey, I'm a high-condition individual, mate with me. The females are choosing males largely based on their condition, which is weakly heritable, but we get a renewal of condition variation, genetic variation in condition in every generation, because condition has a huge mutational target, because so many different traits will affect condition. So this is like classic sexual selection theory. This is textbook stuff again. I'm just saying that so that it's kind of unarguable, at least this part of the thesis that I'm arguing. All right, and so you may have already kind of anticipated what I'm working up to. Evolutionary theory has long recognized the importance of these distributions of mating success in generating sex ratio patterns. So Trivers Willard, for example, first recognized that, hey, if a female, they were thinking of mammals, if a mother can pass her condition across generations, then a high condition mother should create sons rather than daughters, because a high condition mother that creates high condition sons is going to go on to have an extremely high reproductive success, because those sons that are high condition are going to be able to mate with a lot of different females. Whereas a low condition female probably shouldn't make sons, because she's going to make low condition sons that will have 0 reproductive success. This low condition female mother should create low condition daughters, because these daughters are going to be mated with by somebody, and they're going to have some fecundity. And they were thinking of mammals, and it's reasonable to think of passing condition across generations in mammals, because mammals have protracted parental care, lactation, you name it. So you probably can pass condition across generations. So the problem, or a difficulty at least, with Trivers Willard is that there's no clear, or at least undebatable mechanism by which females can manipulate sex easily. It's not as if female mammals can generally just say, hey, I want to make a bunch of sons, and then, muck up her sex chromosomes so that they make sons. Whereas under TSD, it's the environment that influences sex. And so we know that under TSD, incubation, or that under in ectotherms, incubation temperature has a pervasive effect on phenotype. And it probably ultimately affects the health, vigor, quality, and condition of individuals. And we know that this is true for early life traits of reptiles. So we know that incubation temperature affects embryonic survival, embryonic development rate, size at hatching, growth rate, behavior, immune function, post-hatching survival, just to name a few. The list goes on and on and on. Temperature has these huge effects on phenotype in early life. And when we can study later life traits, we also see that it often has an effect there too. So these are for these adult traits that I'm speaking of here are for lizards, because lizards tend to have shorter generation times, so we can actually track the influence of incubation temperature up to adulthood, whereas you can't really easily do that with a turtle that matures at 18 years. But where we're able to track it with lizards, we see that incubation temperature affects physiology and behavior, as well as fitness components, so survival, fecundity, and mating success. And so if incubation temperature affects adult quality, adult condition, then I'd argue that classic evolutionary theory suggests that a sex by environment interaction for fitness is basically unavoidable. And this is specifically how. So let's recast it in the sense of Travers-Willard to make sure that we can understand it graphically. So what classic Travers-Willard is really assuming is that we have sex-specific fitness functions with respect to maternal conditions. So these are expected offspring fitness, I guess. But what they're really assuming is that if we kind of collapse across the sexes, is that there's this simple positive relationship between maternal condition and the expected fitness of her offspring, which is a really simple assumption until you start breaking it down to sex-specific fitness functions. So let's start thinking about the FMF, temperature by sex reaction norm. So let's make a simple assumption with respect to the incubation environment. So let's assume that there's environments that are too hot and stressful and bad, and environments that are too cold and stressful and bad, and there's kind of a Goldilocks environment. So, embryos or populations are adapted to the average incubation environment, whatever that average kind of environment or temperature is. Then there's extreme environments that are bad on either side of that. That's a pretty, I think, reasonable and straightforward assumption. But what this expected fitness relationship with incubation temperature breaks down into is something, when we break it down into something more sex specific, it becomes apparent that males might do an awful lot better in those high condition or kind of Goldilocks environments. So here, what we see is that both male and female fitness are highest in that high quality environment, in the average environment. But it's just that males stand to gain an awful lot more from that high quality environment because they're going to have high quality phenotypes that are going to be able to mate with a number of different females. Whereas female fitness is still highest in that environment, but not nearly as high as that of the males. Whereas the opposite is true in those kind of extreme environments. In the extreme environments that are relatively stressful, females are going to have higher reproductive success than males ultimately, because these males produced under these suboptimal conditions are going to go on to mate with nobody because they'll be low conditioned individuals. Whereas these females that are low conditioned individuals will still be mated with, and they'll still have some fecundity. So the point here is that male fitness is more sensitive to variation in condition than female fitness. And so this particular argument, I think it's been kind of tossed around informally for a long time. I think it was formalized-ish in 2002 by Friedman and Wade. But one thing I don't think, well, probably me and a lot of other people haven't thought a lot about is how we can reconcile that MF pattern. So that's the pattern where we have males at cool temperatures and females at hot temperatures, which is found in all these turtles, or most turtles, with this Travers-Willard idea. And to me, it seems relatively straightforward. So recall that sex is determined by the amount of development that happens kind of above or below the pivotal temperature, or at least it's highly correlated with that. So what that means is that at really low temperatures, they have extremely little influence on sex because very little development is happening. Whereas these high temperatures have a profound effect on sex because development happens so quickly. So it's not beyond the realm of possibility that the selection to maintain these lower these lower pivotal temperatures in this in the FMF species is relatively weak. So these low temperatures are relatively inconsequential to high temperatures with respect to sexual differentiation, and because they have low developmental leverage. And also, I'm going to show you some evidence that these low temperatures have less of an effect on fitness than an equivalent deviation into to warmer temperatures. So that's how I think we can just reconcile the MF pattern with this Travers-Willard idea. It's just the idea that these low temperatures have low developmental leverage and don't actually influence sex very much. And so they just be lost. All right, so I've kind of made that pitch, I've made that argument. Do I have any evidence? Well, I do. So this stuff isn't published yet, and there's nothing kind of more motivating than knowing that you have to give a talk to some of your colleagues to kind of organize your thoughts and start working on some of the stuff to get it out there. So here's the evidence. What I've just argued really is that these pivotal temperatures evolve so that male production is matched with good thermal environments, with high quality phenotypes. So these pivotal temperatures are locally adapted, and they should be strongly linked with thermal stress, so we can kind of use them to delimit what is a thermally stressful environment and what is not a thermally stressful environment, at least to some extent. And I'll expand on that here. So I had an undergraduate student, Lauren Lawson, who did a wonderful job on this. And in fact, Melanie helped in the very initial stages of this experiment too. Test some of the predictions that arise from this Travers-Willard idea. So I'll direct you to this graph. Is my mouse pointing? Yeah, this graph down here. So here I'm going to show embryonic mortality on the y-axis in relation to incubation temperature. So embryonic mortality would be kind of, if we have different clutches of eggs incubated at different temperatures, embryonic mortality would be the proportion of eggs in that clutch that died, which should be kind of correlated with thermal stress. So let's assume that 27 degrees Celsius is the pivotal temperature. So above that temperature, we're going to get females, and below that temperature, we're going to get males. Let's just think about MF species for now. So that's males are produced at cool temperatures and females are produced at warmer temperatures. So now, because we're going to involve a lot of different species of TSD species in this experiment, what we want to do is we want to center our mortality data with respect to species-specific pivotal temperatures. So if 27 degrees Celsius is the pivotal temperature for this hypothetical species, it suddenly becomes a 0, and then we have deviation from pivotal temperature, so plus 6 degrees, minus 6 degrees. So in that way, every species that we incorporate into our study will be centered in a similar way or the same way. So we extracted incubation temperature and mortality data for turtles from a database. So this is across studies done over the last 30, 40 years. We found species-specific pivotal temperatures in the literature, and where we needed to, we substituted the pivotal temperature that wasn't available for that population or species with one from the closest geographic location. We ended up with 29 studies, 202 mortality data points, 15 turtle species that represented 43% of turtle families. We only had one species of FMF, snapping turtles, that was represented in 6 studies. So what I'm going to do here is I'm going to show you the results where we analyze the FMF species together with the MF species, and then just the MF species. And I'm doing this mortality with respect to pivotal temperature. So for these FMF species, we're just looking at the upper pivotal temperature, and we're ignoring that lower pivotal temperature. We're pretending it doesn't exist. So what do we expect to see in these data, in these mortality data that are centered on pivotal temperatures? Well, critically, what we think should happen is that mortality should accumulate with temperature a lot more quickly above the pivotal temperatures compared to below pivotal temperatures. If below the pivotal temperature is generally less stressful, then mortality should accumulate relatively slowly compared to above it. So put in a different way, if we kind of rescale the variable here and we have absolute deviation from TPIV, so in other words, this minus 6 becomes a plus 6, we just kind of put it on a separate curve, and we're looking at how mortality accumulates above the TPIV versus below the TPIV with distance from TPIV, we expect that mortality is going to be accumulate faster above the pivotal temperature. All right, so let's explore the data a little bit. Remember, this is an undergraduate study. This is a pretty cool undergraduate study. So on this graph, we have clutch mortality on the y-axis, and we have deviation from incubation temperature on the x-axis. Each color in the graph is a different study, and the size of the point represents the number of eggs in the clutch with which mortality was estimated. So we're putting more weight on clutches that were bigger, because we're more certain that proportion reflects the amount of mortality actually occurring at that temperature. So with respect to combining the MF and FMF species into one big graph, we see this approximately parabolic relationship with the lowest embryonic mortality around TPIV and probably just below TPIV too. And the same is more or less true when we just look at the MF species together. So the further you get away from TPIV on either side, the worse things get. But it seems like it might be especially the case above TPIV. Where, of course, even though I'm arguing that cool temperatures are less stressful than warm temperatures, I think it's reasonable to expect eventually get high mortality at cool temperatures because we're holding them there at constant temperatures. These aren't fluctuating temperature experiments. These are constant temperature experiments, which is essentially an evolutionarily novel environment, right? So holding eggs under like 10 degrees Celsius for 3 months is probably going to eventually kill them. But anyway, just to kind of point out why we might have mortality at cool temperatures too. So the key test here, though, is to test this prediction. Is mortality accumulating faster above TPIV than below TPIV? And this is what the data look like for the MF and FMF species combined. And you can see that in red here, we have absolute deviation from pivotal temperatures above TPIV. And you can see that mortality is accumulating a lot faster, rising a lot faster with temperature than below TPIV. And that's more or less the case as well with when we just look at the MF species. It's pretty messy data. Keep in mind, we're just combining various different experiments here with a random effect for study. But it does seem to support the main hypothesis, the main prediction. The other cool thing about this study is that when you look at the clutch mortality, clutches that are more male biased had higher survival, so lower mortality, than clutches that were female biased, as you'd expect from if females are produced under environments that are relatively stressful. And assuming that there's no kind of sex biased mortality within the clutch or something like that, which I don't think is an issue. But when you put sex and temperature into the same model, sex disappears as an explanatory variable, and it's just temperature doing the explaining, which is what you'd predict under our hypothesis. Okay, so we have here several lines of evidence that point towards female producing temperatures being relatively stressful. But all we've done here is just show the early life environment. Remember, turtles and crocodilians live for like decades and decades, and some of them live for like over 100 years. So we might want some evidence of some long-term effects. Of course, you can't really study this very easily by marking, turtles and follow them for, 100 years. Not only that, but turtles have extremely low survival in early life, so it's kind of hard to get at this question in the first place. So we have to get at it indirectly. So what should happen with sex ratios under this Trivers-Willard idea? So let's imagine that we're right and that we have these environments that produce high condition juveniles and high condition offspring, and we have environments that produce low condition offspring. What should be happening is that we should have sex biased mortality. The individuals, females, I'd argue, produced in the poorer conditioned environments should die at a higher rate than males. So what the result should be that relative to birth sex ratios, adult sex ratios should be more male biased because males are produced in the high quality environments. But the prediction actually gets better than that. There's some theory on this that was done by Bull and Charnoff in 1988, and a different subset of theory done by Friedberg and Wade in 2003 that show that if this Trivers-Willard idea is right, then what we'd expect is not just any sex ratio at birth. We specifically expect female biased sex ratios at birth. And it gets really mathy, and I'm not even sure I fully understand it yet, but let's just take their word for it. These are, theoreticians. So what we would expect under Trivers-Willard is female biased sex ratio at birth, followed by a gradual followed by more male biased sex ratios at adulthood. One of the things I've argued to date is that we have different adaptive explanations for TSD in these major clades. And so what we might expect then is that for lizards and the tuatara, we might expect a totally different pattern. It might look like this. It might look like this. I mean, it doesn't really matter what it looks like. On the balance of probabilities, it's probably not going to look like the pattern that we see in turtles and in crocodilians. It could, but probably not. So let's look at the data. So again, there's nothing more motivating than to do some research and reading than knowing that you have to give a seminar in a couple of weeks. So I found these data about a week and a half ago to support this idea. So this is a study published in 2019 on sex ratios in reptilian sex ratios in BMC evolutionary biology. They didn't analyze the data with respect to this question, so I'm kind of presenting it initially here. So what do we see? We see that in naturally produced populations, so wild sex ratios, at birth, at hatching in turtles and crocodilians, they are in fact female biased. And we have a decent sample size here. We have 43 different populations of turtles and 11 different populations of crocodiles. And then for better or for worse, by adulthood at least, we do have these more male biased sex ratios with, again, not super terrible sample sizes, especially for turtles. I haven't done this in a phylogenetically explicit context yet. I need to learn how to use GLLM or something to do that, which sounds really difficult, and it's a summer project. So these are just arithmetic means, but they are promising. They do point in the right direction. So what about squamates and the tuatara? Well, we have very small sample sizes, especially for birth, but it does seem as though they're a bit higher than you might expect if it's kind of the same, if they follow the same pattern as turtles and crocodilians. So that's cool. It kind of fits the what we'd expect under Travers-Willard, at least in terms of these arithmetic means. And this, to me, this last slide on the adaptive significance of TST is the coolest part. So what I've argued is that TST evolved to match males with high quality environments. And if that's right, then the reason TST exists is because temperature specifically is just an environmental variable that happens to have this massive influence on lifetime condition, on the quality of individuals. But a bold prediction under this idea then is that it won't just be temperature that influences sex. Any environmental variable that's consistently in the embryonic environment that has an effect on condition should influence sex, maybe roughly in proportion to how much of an effect on condition it has, right? Carbon dioxide influences environmental sex determination in 2 species of turtles. This study has been cited about 2 times, and the study was published in 1994. I suspect it's because we can't make sense of what's going on here. But thinking about it in the Travers-Willard context, I think it might make sense. So what they did, this is another reason that it's a really cool study, and I'm glad they did it. It's not replicated in the sense that some other experiments are, but it still has some pretty cool findings. So we have, they're manipulating carbon dioxide and oxygen somewhat simultaneously. So here we have mortality of embryonic Trachomas scripta. So that's your pet store turtle, kind of increasing as you increase carbon dioxide. So it goes from 0% mortality at kind of normoxic conditions. And by the way, carbon dioxide in the atmosphere is about 0.04% normally. So it goes from 0% mortality up to about 100% mortality once you start manipulating carbon dioxide and oxygen at the same time. But even when you don't manipulate oxygen, you still have 15% mortality. And so this is environmentally relevant because all embryos in all reptile nests respire. And especially in turtle nests, they're subterranean nests, and they're kind of in soil that's not necessarily porous. It's sometimes very moist. It can be anoxic, and there might not be an awful lot of air exchange. So carbon dioxide poisoning or high levels of carbon dioxide are probably very common in turtle nests. So this is a pervasive environmental phenomenon that they're looking at. And at the same time that mortality increased, they're incubating all of the eggs at 27 degrees Celsius here across all treatments, which is close to this species or this population's pivotal temperature. At the same time that mortality increased, we saw this massive decline in the number of males produced. So at normoxic conditions, we had 24 eggs that hatched out of 24 or 25, I think. They were almost all male. They were able to sex 7 of the embryos that kind of died under this treatment over here. They were mostly female. And this is without manipulating temperature. So they didn't replicate it in an experimental way. They replicated it across species. This is the Mississippi map turtle done in the same study. They just did 2 treatments here because they had fewer eggs, but you can see that indeed you're getting this increase in mortality followed by or concomitant with an increase in female bias in the sex ratio when you crank up carbon dioxide. I'm not going to go into any particular studies to say this, but there's also some evidence that moisture in a nest is associated with high quality phenotypes. So when you have more moisture, these male or the offspring are a little bit more robust, and high quality environments, or sorry, high moisture environments Some papers have tried to show that they're also associated with more males. They're a little bit less convincing, but there's some evidence there as well. But what the cool thing is that what this means is that it's not really temperature dependent sex determination. We have to kind of rethink the way we're thinking about this. It's environmental sex determination that matches males with high quality environments. Temperature is just kind of this pervasive actor in it because it's important, but it's not the only actor. So the summary, I'd argue, is that just Trivers-Willard provides a general reason for a sex by temperature interaction for fitness among turtles and crocodilians, and I've presented some evidence to support the idea. And we can leverage this Trivers-Willard idea to understand what delimits thermal stress in some of these environments by looking at these T-PIVs, because these T-PIVs under this hypothesis should delimit stressful versus non-stressful environments. And it also allows us to kind of rethink what temperature-dependent sex determination is in the first place, which I think is super cool. And it might have some relevance to climate change, which is what I'm going to talk about next. I only have about 5 minutes or so left, 5, 6, 7, 8 minutes. So, yeah, I'll be able to get through this last little bit. So this is a depiction of the global temperatures since that massive asteroid hit us about 65 million years ago. So the temperatures plummeted, and we're over here now in around 2005 or so. And so there was a massive kind of global decline in temperature that occurred over millions of years. And obviously there's a lot of fluctuation in there that might be important as well. And so a study came out recently that showed that, or that suggested that TSD survived this KPG event, this asteroid impact, better than species with genetic sex determination. They kind of reconstructed ancestral states of various different species and inferred TSD versus not TSD, which it was really interesting. But whether or not you believe it, the problem is still here, that we have this massive climatic fluctuation over time, and TSD species still persist. And so how do they persist? The short answer is we don't know. And evolution is, of course, an option, although when we're talking about things like alligators and snapping turtles, their generation time is extremely long, and the heritability of these TSD traits is extremely low. The visibility to selection of the T-PIVs, for example, is extremely low. So these are traits that do not evolve rapidly, apparently. So we don't know why specifically they've persisted through climate change, but we may be able to find some clues in our long-term data from up in Algonquin Park. We've been studying snapping turtles up here for almost 50 years, and we have some long-term sex ratio data that is, of course, not published yet. The life of an assistant professor with kids. So there's Algonquin Park, and it's in southern Canada, but it's cold up there. So here we have a high altitude environment, at least for Ontario, 440 meters. And we have these short, cool growing seasons, these really thermally constrained growing seasons. This is turtle V3 putting around in an early May snowstorm. So they're at their climatic range limit. The climate here in Algonquin Park is about the same as the climate like 150 or odd kilometers north, just because of that high elevation in the Algonquin Dome. And why might climactic range margins matter, especially in the north? Well, I've misappropriated this data from a recent study in BMC evolutionary biology. It's not exactly the type of data you'd want to answer to uphold the argument I'm about to make, but it's similar. So here we have 85 reptile species. We have the mean temperature of the breeding season for these reptile species. So that's the temperature over which the eggs are incubating on average in the species range. And we have an index of annual climatic variation. So how much temperature is changing from year to year to year. And the cooler your breeding season, which should be pretty well correlated with latitude, so higher latitudes should have cooler breeding seasons, the more interannual variation in temperature you have. And why does that matter? Well, any kind of variation is good under climate change, because the argument under climate change is that we're going to homogenize these sex ratios, and they're all going to become one sex. So more variation, at least in the near term, is good for maintaining variation in sex ratios. So these climactic range margin populations might be important. But intuitively, you can also imagine that a species with this ancestral type of TSD, this FMF pattern, might produce more variable sex ratios, because here we have temperature varying over 2 T-PIVs within a season, right? Rather than just having the one kind of T-PIV and doing this. All right, so has climate changed in Algonquin Park? It has. These average temperatures from the 70s and 80s to now, and you can see that we have warmer summers and warmer falls, but not necessarily warmer springs. All of our study, all of our sex ratio stuff, if you can see my, I am assuming you guys can see my arrow, it takes place on this 100-meter long dam at the very south side of Saskatchewan Lake here, in the south side of the picture as well. So it's 100 meters long by 10 meters wide, and that's very important in a couple slides. It's this tiny little area, human-built area. So from 1982 to 1998, when the turtles, we've been doing this since 72, but specifically for the sex ratios from 82 to 98, when the turtles came up to nest on the Lake Saskatchewan Dam from Lake Saskatchewan and the surrounding areas, because the dam attracts them from tens of kilometers away. We would dig up their eggs, measure their eggs, and rebury their eggs with temperature data loggers and measure the females and so on and so forth. So the sex ratio study went from 82 to 98, and then it started again when I started in 2016, and it's been going to the present day. So this is probably the longest, one of the longest sex ratio studies on turtles or on anything in the world, and it's the only study on a type 2 species that I know of. And we can ask the questions, are sex ratios resilient to climate change, or have they changed over several decades? And so this is what 20 years of data collection and a boring result looks like. I'm being facetious. There's actually a lot to this, but on the face of it, there's nothing going on. So we have the average sex ratio on the dam. So this represents the arithmetic mean of a whole bunch of different sex ratios and a whole bunch of different nests on the dam. So that's the arithmetic mean for that year. So the mean sex ratio is not changing over time. And so one way they might be doing this, in spite of having kind of warmer summers, is that, well, maybe they're just nesting earlier so that they're keeping their eggs at a relatively constant incubation temperature, just because they're offsetting, they're nesting earlier, right? And there's some, I'll just skip over this slide, but there's some theory to support that. So what do we see when we look at long-term nesting trends in the snapping turtles in the dam? Well, again, this is mean nesting date with respect to time, and nothing. There's no relationship between nesting date and time. So they're not nesting earlier to kind of keep their nests cooler. So how is it that despite this kind of warming that we're seeing in the summer, which we've made some models on this, and this warming is occurring right during the thermosensitive period, the average thermosensitive period, how are they avoiding this? Well, this is one possible reason. So this is the sex ratio of individual nests on the dam from 82 just to 98, because I didn't have the recent data, but so this is the sex ratio for all nests where we've sexed more than 10 eggs in the nest out of an average of 35 or 40 eggs or so. So we have a decent idea of the sex ratio in the nest for each one of these data points. And you can see that within a given year, in an area that's 100 meters long by about 10 meters wide, and they only nest over a period of about 2 weeks at most, usually like 10 days. So there's only 10 days difference in nesting times maximum between individuals. We're seeing this massive variation in sex ratio. The microclimatic variation with respect to temperature on this dam must be enormous. Or maybe it's not just temperature influencing sex. Maybe this explains why people aren't able to construct accurate models of temperature-dependent sex determination, right? Maybe there's other things going on within the nest that affect sex, like hormonal factors, carbon dioxide, moisture, you name it, as I described under the previous hypothesis there. All right, so I think I'm out of time. Luckily, I'm also on my last slide. I'm just going to wrap up and say that, yeah, I believe that we're seeing good evidence of climate change resilience up in Algonquin Park. I didn't present a convincing case of it because I wanted to make a better case with the adaptive thing, but I do believe we have evidence of this. We have extensive among female variation in nest sex ratios within years, and also moderate among year variation in mean sex ratios. So the mean sex ratio of nests ranged from 15 to 65% males across all years, which is a fairly big range. And the other thing that might be going on, as I described in the earlier section, is that sex might be determined not just by temperature. There's a variety of other factors that could be involved that are either indirectly or directly linked to that Travers-Willard hypothesis that I described. So that's my story, and I'm sticking to it. So yeah, thanks to Melanie and Boris for inviting me, and thanks to a number, especially all of the students that collected data over all of those years. And I kind of summarized that in, like, this is what a boring result looks like. Well, really, a ton of actual work went into that. I was just being a bit facetious. So those students are absolutely wonderful for doing that. And I've had wonderful graduate and undergraduate students in my lab, and they're also responsible for a lot of the ideas and data here as well. So thanks so much.